All right, I'm Michael Fears. I'm going to talk to you today about molecular motors and cell signaling. This is a project that I work on. I work for Dr. Josh Baker, and we work in conjunction with the lab down the hall with Mariam Ba and Maria Valenchek. So I'm going to give you a brief introduction, some background on muscle structure and myosin protein function. And then I'm going to go into my current work and talk about micropatterning techniques, polyacrylamide gel surfaces, and some of my preliminary results I've gotten so far, and talk to you about some of the future directions of our project. So our overall goal of the project is to study how the mechanics and kinetics of actin-myosin interactions, how they affect cell signaling. And we're interested in this down here, the inside-out force generated by actin-myosin. So in active muscle, you get a stretch, and then you get this contraction here. And so we're interested in how that contraction and the force outputted by that contraction leads to signaling outside this cell. So one of our goals is to create a model system for studying these mechanics in an in vitro system, so not in a live animal, and then how to perturb the myosin mechanics and then observe the changes in protein localization that signal to the cell's environment. So I've talked to you a little bit about this before, about hypertrophic cardiomyopathies, and it is when you have an enlargement of the left ventricle of the heart. So in a healthy heart, your left ventricle looks like this. In a hypertrophic heart, due to disease, which comes from pressure volume overload, this can be from health factors, environmental factors, or in this case, genetic mutations. It leads to this hypertrophic condition where you have a thickening of the wall and a decreased volume in your left ventricle. And why this is kind of of interest to us is because the fact that these genetic mutations that are mostly in myosin or in other sarcomeric proteins lead to this disease is strong evidence that this inside-out signaling from the cells plays a role in disease as well as normal function in the heart. So there's been many studies already on the pathways that lead to hypertrophy from like the signaling, the outside-in signaling pathways. And this figure is simply just to illustrate how complex it is, how many pathways there are. We don't study any of these. But very few people study kind of the reverse of this, which is what we're looking into, is how when you change the mechanics or force output of the cell, what is the result in these pathways the other way that leads to signals that are sent to neighboring cells and tissues. Why is it important to study mechanotransduction like this? Well, a lot of these underlying mechanisms of how this force is transferred from the sarcomere to neighboring cells isn't really understood. So if we have a better understanding of it, it gives us more targets for disease therapy or improving treatments of these diseases, as well as cardiac tissue remodeling, biomaterials, advances in tissue engineering, things like that. All right, so now I'm going to go into the background a little bit about myosin function and muscle structure. So up here is what a kind of a graphic of what a myosin looks like. You have two heads with the tail that intertwine to make this filament. Myosin are responsible for a wide range of functions in cells for directed movement of molecules, subcellular components like organelles, whole cells themselves, whole tissues, and of course whole organisms like us. Actin and myosin are what move your muscles and allow you to not sit in one place. Myosin is a motor that depends on ATP, so it converts chemical to mechanical energy by utilizing the energy that's released when you hydrolyze nucleotide triphosphates like ADP or GTP. So this is a much better diagram than the one I tried to draw on the board for you before, showing how muscle is structured. So from muscle, it comes down to made up of muscle bundles of fibers. And then each fiber, in the end, comes down to a myofibril. and each myofibril is made up of a sarcomere. Sarcomere is kind of the basic unit that repeats throughout these muscles. And within the sarcomere, here's your actin filaments, and then your myosin filaments along the edge here, so that myosin binds actin and then slides against it, which is why you get the shortening as it pulls these actin filaments in this way. So like I said, myosins are actin-based proteins. The hydrolysis of ATP in the myosin head results in a conformational change in that myosin, so it actually swings, and that when that neck swings and pulls on the actin filament, that's what results in the muscle contraction. And the contraction itself is a net result of many actin-myosin interaction filaments sliding relative to one another. And the result is your muscle fiber shortens. All right, so I'm going to take you through the kind of the 4 steps of the actin-myosin interaction that lead to a contraction. So the first, you start here at what's called the Riger complex, and it's the point, this is your myosin, this is your actin filament. So the Riger complex is myosin bound to actin with no nucleotide bound, no ATP. And this is what we call tight binding, or the Riger complex. You guys have heard of rigor mortis. When someone dies, you know, the body gets stiff. That's where this name comes from, the Riger complex, because if you're no longer, you know, in a dead body, you're no longer producing ATP, so there's no nucleotide to replace this. So that's why that the muscles bind and no longer want to move anymore. So from this first step, from your Riger complex, when ATP binds to this, the actin and myosin actually dissociate, and this is your unbound step. Then the myosin hydrolyzes the ATP to give you ADP-PI complex, and this is a weakly bound step. So at this point, it's weakly associated with myosin. You not have a strong bond here, and then the transition from 3 to 4 is what we call the power stroke. So it's actually when this phosphate leaves the complex that you get the myosin head cocking, and you get what we call the power stroke, and this is your strong binding interaction. And then once the ADP leaves, it goes back to this Riger complex, and the whole cycle starts over. In physiological conditions, this step here is very brief or kind of non-existent. There's a lot of actual intermediates in here, and people argue all the time over the little intermediate steps between these and which ones are responsible for the different roles of the contraction. But like I said, a lot of that is controversial just because we don't have a lot of good structural information for these filaments in their natural states. All right, so mechanical transduction is the transduction of forces in the cell that to some other signaling. So a force will be sensed by the cell, and it will result in an actual signal in the cell. So your cells are constantly exposed to mechanical stresses. You have fluid shear stresses on cells that are in arteries or other linings in the body. There's environmental factors that exert forces on the cells, and of course cell-to-cell interactions from one cell next to each other that will pull on another. So mechanical transduction is simply the process by which cells sense these forces and translate them to an actual physiological response. So focal adhesions are are complex of proteins in the cell that actually connect the cell, the inside of the cell, to its outside environment. So this is an actin filament. This would be the cytoskeleton of the cell. And it connects through all these adapter proteins to integrins. And integrins are kind of the key here because they're the transmembrane proteins that bind these and link to the actin filaments as well as to the extracellular matrix. So when force is exerted in the cell, when it contracts, it's translated through these proteins to the outside of the cell. So you can kind of think of these proteins acting almost as springs. You pull on one and it's going to pull on the next one and transduce that force this way or the other way from outside in. And the integrins are kind of important because it's the end result of that pathway. The more, this complex will start simply as one integrin and a couple of these proteins connected to the actin filament, and then as it develops, because of forces pulling on it, it'll recruit more and more integrins until you get this complex, and this will grow and grow. And it's kind of dependent upon the amount of force that's being applied at that focal adhesion. All right, so now I'm going to talk to you a little more about my current work, talking about micropatterning techniques, our polyacrylamide gel surfaces and why we use them, and some of the results we've gotten so far. So a big part of this project was just developing this model system. It's to study these integrin signaling and force mechanisms in a cell. We want it to be as close as possible to what's actually happening in a human body. Now, whenever you take these cells outside of a body and plate them, their phenotype changes. They don't look the same. They don't behave the same as they do in vivo. So our goal is to maintain as close to in vivo conditions as possible, but still have an in vitro system. So a big part of this is the substrate stiffness that you grow these cells on. The stiffness of that substrate will affect what the cells look like, how they develop, and it's actually pretty interesting. A lot of work has been done with stem cells, and when you grow stem cells on different substrate materials, they actually will differentiate into different types. So on a really stiff material, it will turn into like a muscle or a bone progenitor cell. On intermediate stiffnesses around Young's modulus of 11 kilopascals is similar to muscle tissue. And softer than that, you get soft tissues. So this is work that was done by a group led by Engler. And this is cardiac myocytes that were grown on different stiffness substrates. And as you can see, when you go from soft to more stiff, you see changes in the cell. And this here is what you kind of want to see. You see those green dots that start to form. It's not this big, long stress fiber. That's what we call striations, and that's that pattern of sarcomeres that you actually see in a normal muscle cell. So as you can see, the stiffness definitely affects how these cells grow. So our goal is to shoot for stiffness around 11 to 12 kilopascals so that we maintain that physiological range. So we do this by growing the cells on a polyacrylamide-coated surface, because the polyacrylamide gives us control over the stiffness of that substrate. And it's great to use because it's optically clear, so that we can image through it on the microscope, and we can embed things like fluorescent beads in that layer so that we can monitor the movement of the cells without having to put markers in the cells themselves. So this is an example of why we pattern the cells. So if you were to just take these cells and throw them in a culture dish after you isolate them from an animal, this is what they'd look like. They just kind of spread out and look like a mess. This is tissue samples that are taken from an actual adult heart. And as you can see here, the cells don't look like this. They're aligned, they have organizations. So when you pattern the cells, when you direct their growth on the gel, you get something like this, which looks a lot more like what you would actually see in the tissue and not just cells cultured on a dish. So we combine these two techniques of using a polyacrylamide gel and micropatterning. So we have a photoresist that we've created that has these ridges on it, and these are about, the spacing here is about, depending on which part of it, is 10 to 20 microns. And we cast a PDMS, what we call stamp, polydimethyl siloxane stamp, on that. So when you take it off, you're left with those ridges. We then incubate that with a protein, an extracellular matrix protein. We use laminin, and that's a protein that's found in the an extracellular matrix of your tissues, which cells bind to and kind of hold in place. So once you rinse that off, you're left with your protein on top of your patterned surface here. You then take an acrylamide gel, and those little green dots represent the fluorescent beads that are inside. And then you activate the surface with a cross-linker. And then, so when you apply this stamp to your gel, the cross-linker binds to your basement protein, and you're left with a patterned gel. You then take your isolated cells, throw them on, and it takes about 6 to 8 hours, and they'll attach. And then once they attach, they'll grow and spread out into these lines. So this is just a light microscope image of cells that we've patterned here. And then this image here, the green that you see is the nucleus. These little dots on the side are the microspheres we've embedded, and it's kind of hard to see in this light, but they've been stained for alpha-actinin so that you can see the green, the organization of the fibers as the cells grow end-to-end and mimic an actual fiber you'd find in the heart. So is this movie working? Okay. So what we do is we culture these cells, and you can see it's moving slowly here. So then we put them on the microscope, and we actually stimulate them to contract. So we apply a voltage of about 4.5 volts, and we pace them at 1 hertz frequency. So as the cells beat, you can see the beads move in response. So when you process this image, and we use this software called Fiesta. It's an acronym, and it was developed by a group led by Michael Dietz in Germany. And it gives you nanometer resolution of these beads. So you get this displacement. So from one of these beads, you get this track like this. So the next step is we throw on an inhibitor, blebistatin. It's a small molecule inhibitor, and it slows down the phosphate release from myosin. So it's not actually interfering with the binding of myosin actin, it's just changing the kinetics of that reaction. So as you increase your concentration, the amplitude of your contractions start to decrease until they've gone away. This is kind of where we're at now. The future prospects is to look at how, when you add blebistatin, how it's going to change the signaling. And what we hope to see, so this is Estelle, the red is stained just for integrins. The green is a talin GFP protein, and the right is the co-localization of these two. So you can see that they're, you get the talin in the same area, the orange color, in the same area as the integrin. So what we hope to see is that as we add blebistatin, we should see a decrease in the amount of co-localization we see here. This is where we're at now. It's actually really difficult to get these cells to express the virus that we used that has the fusion protein. It's really hard to get muscle cells to express, so this has been a bigger challenge than we thought right now. And then what we'd like to do after this is go one step further and look at how calcium handling in the cells affects force generation, because the muscle contractions are overall regulated by calcium. So from here, we'd like to do calcium imaging. You load the cell with a calcium-sensitive dye, and you can actually image the calcium transients as the cells contract. So in summary, we talked about cardiac myopathies and how they develop from altered force output in cardiac muscle. Talked about muscle organization and how myosin produces mechanical energy from chemical energy. I've gone over some micropatterning techniques that we use in our work and talked about how we use blebistatin to inhibit force. Is there any questions? Oh, sorry, he had his hand up first.
Last time you mentioned that athletes are susceptible to the hypertrophy that you're talking about.
I didn't mean that they were susceptible. I was just kind of using it as an example because I know a lot of people have heard of that when you have a young athlete, someone who's healthy, you know, just having heart disease or dying from cardiac arrest. So I'm not saying that they're susceptible. It was just an example that I used.
Okay. Well, I guess I'll still ask my question.
Yeah.
If they stop, you mentioned that the mechanism that makes hypertrophy is the same that builds muscles in the rest of your body, right?
Okay.
Okay, I don't think this is going to work out. But, if you stop being athletic and subjecting yourself to the pressure and volume overloads, will it reverse itself and atrophy like muscles in the rest of your body?
It won't reverse itself, and that's what's kind of particular about this why we're interested in this inside-out signaling is because of the fact that it's actually a mutation in the myosin that they're suffering from, and that mutation is affecting the interaction with actin and the actual force output from the muscle motors. They'll lead to that disease state anyways. In severe forms of this disease state with certain mutations, 50% of the people don't even live past the age of like 45, regardless of health, lifestyle, things like that.
So the athletes that are susceptible also have these other genetic and...
Right. Most of the time when it turns out they have a mutation in myosin or some other sarcomeric protein that affects force output. Yes?
I don't know much about this, but the stiffness of the polymer you were using, how did you measure the 11 kilopascal?
I didn't.
Point test, or?
I didn't measure it myself. A lot of people have measured it. They actually use AFM to measure it is the most accurate way. You can actually measure it yourself crudely, and it's really close to an AFM measurement just by making like a chunk of the material and then applying a known weight to it and measuring that the change in length. And you can determine it that way, and it's actually a really, really good estimate of what it's at. But I've pulled data from other people that have published, curves showing the concentrations of the acrylamide to the bisacrylamide and the what stiffness you get. So I've just used other people's data for that. Any more questions?
That time was 1845.
Yeah, I went over.
We have 7 minutes.
Okay, we don't have time to do
Oh, well.
Saved by the Bell. But, okay, so I'd like you to